Hello, so today we're going to talk about the Watson and Crick model of DNA. So first let's look at the overall structure of DNA. DNA is a helical structure consisting of two anti-parallel complementary strands. Anti-parallel means that the strands go in opposite directions. Complementary means that the pieces of the strands, the nucleotides, pair together. We'll talk more about these concepts more specifically in a second. So since DNA is helical, there's a handedness to it, meaning it spirals up in one direction. Generally in nature, DNA is right-handed. However, left-handed DNA has been produced in a lab setting. DNA also has major and minor grooves. These occur where it's pinched. The major grooves are where there are large gaps, and the minor grooves are where there are small ones. In the second image, we can see what the two strands look like flattened out. Each strand is a collection of repeated units known as nucleotides. One important structural note is base stacking. This is when the flat base pairs in the center stack on one another. This increases the hydrophobic interactions in the center of the molecule and provides stability. So now let's look at the nucleotides in a little more detail. In this image, we can see that a nucleotide contains three things: a phosphate group, a deoxyribose sugar, and a nitrogenous base. The sugar and phosphate molecules bond to give the strand its structure. The deoxyribose sugar has 5 carbons. The second carbon is what makes it a deoxyribose molecule, as it's missing an oxygen next to one of the hydrogens. If there was an OH group on the 2 carbon, it would be a ribose sugar. The 3 and 5 carbons are where the sugar and phosphate bind together. The phosphate group is located on the 5 carbon and bonds to the next nucleotide's 3 carbon. This is what gives the strands their directionality and makes them anti-parallel. The nitrogenous bases bond to the 1 carbon. Finally, the nitrogenous bases can be either purines, such as adenine and guanine, or pyrimidines, such as cytosine and thymine. The nucleotides are complementary. The adenine pairs with thymine, while guanine pairs with cytosine to create complementary strands. This pairing occurs via hydrogen bonding. So now let's have some fun with the problem. Hey, Zach, I need your help. I want to raise the melting temperature of some DNA in my lab project. What can I do? Well, Jimmy, here's one idea. The phosphate groups on the outside of the strands have negative charges. This polarity causes two strands of DNA to repel from each other. Luckily, the DNA molecule has other charges that can keep them together. So why don't we put the DNA in a solution of high concentration saline sodium citrate? Because see, the salt solution has a positive pH, and this will neutralize a lot of the negative charge of the phosphate backbone. This actually will stabilize the DNA structure, allowing its attractive forces to overpower the repelling force of the phosphate more than usual. Oh, so putting the DNA in this buffer stabilizes it, then it will need a higher temperature to pull apart the strands and denature the molecule. Yeah, you got it, Jimmy. Why don't you give that a try, kiddo?